The hepatitis B virus is classified in the realm Riboviria, which uses RNA-dependent polymerase for replication. It belongs to kingdom Paranavirinae, which has a sole phylum Artvirviriculta, which has a sole class Redtraversites. This class includes viruses that encodes RNA-dependent DNA polymerase, also known as reverse transcriptase. The class Retroviricites has two orders, one of which is the order Blueberviralis and its sister order Ortberviralis, which contains all RNA viruses that replicate through a DNA intermediate and DNA viruses that replicate through an RNA intermediate, except the family Hepatnaviridae. The hepatitis B virus is classified in the family Hepatnaviridae. These are small envelope viruses with partially double-stranded DNA of approximately 3,200 bases. It expresses three major sets of proteins, which are the pre-core or core proteins, the polymerase proteins, and the pre-S and S proteins. The hepatitis B virus is classified in the genus Orthohepatnavirus, which has humans and mammals as its natural host. The hepatitis B virus has a species name, which is the hepatitis B virus but in italic form. It is the etiological agent of the hepatitis B disease, which causes liver complications. The Baltimore classification of the hepatitis B virus is class 7. These viruses uses double-stranded DNA and replicates through an RNA intermediate with the help of an RNA-dependent DNA polymerase or reverse transcriptase.
The HBV variant, also known as plain particle, has a spherical double shell structure with a diameter of 42 nanometers. Its lipid envelope contains surface protein projections called the hepatitis B surface antigen, which is arbitrarily designated as large, medium, and small. This lipid envelope encloses an icosahedral nucleocapsid, which is a result of the self-assembly of hepatitis B core antigen. Floating in the space between the surface and the core of the HBV, you would find another antigen known as the hepatitis B E antigen, which is released or escapes during the replication of the virus. The icosahedral structure packages a circular, partially double-stranded genome, about 3.2 kilobase pairs, in a virally encoded polymerase that is covalently attached to the 5' end of the antisense strand.
Genome organization of HBV. HBV genome are known to be relatively small and have overlapping coding regions and regulatory elements. They have a positive sense strand and a negative sense strand DNA. So the red here represents the positive sense strand and the blue represents the negative sense strand. The HBV genome has four promoters, namely pre-S1, pre-S2, P-core/E, and PX. Each promoters are divided into two, the liver-specific and non-tissue-specific promoters. Pre-S1 and P-core/E are liver-specific, while the remaining two are non-tissue-specific. Promoters indicate where DNA transcription starts. They are important in gene expression because they control the interaction of RNA polymerase with the DNA. HBV genome are enhancers, DNH1 and DNH2, but GRE is considered as an enhancer. Therefore, there are three total enhancers in the HBV genome. Enhancers from the word enhance enhances the chance of transcription of a specific gene to occur. Enhancers increases the possibility of transcription by presence of transcription factors. DR1 and DR2 are 11-base direct repeat sequences which are regulatory elements of the HBV genome. DR1 can be found in the HBV RNA, while DR2 is found in the negative sense strand DNA of HBV. These direct repeat sequences help the virus integrate to another DNA. The polyadenylation signal is important in the synthesis of mRNA. It is considered as a regulatory element found in the HBV mRNA.
The World Health Organization recognizes 4 methods for inactivating the hepatitis B virus. First, the acid pH inactivation, heat treatment, solvent detergent inactivation, and UV radiation. In acid pH inactivation, the exposure of virus to low pH denatures its target proteins. Hepatitis V virus is inactivated by exposure to sodium hypochlorite or 500 mg of fluchlorine per liter for 10 minutes, and 2% aqueous glutaraldehyde at room temperature for 5 minutes. The heat treatment uses heat to denature the target protein and viral impurity. For the hepatitis B virus, heat treatment at 98 degrees Celsius for 2 minutes is effective in inactivating the virus. The solvent detergent inactivation. This process removes or inactivates the lipid coating of the virus. Solvents that are effective in inactivating the virus are sporicidin at pH 7.9, formaldehyde at 18.5 grams per liter or 5% formalin in water, 70% isopropyl alcohol, and 80% ethyl alcohol at 11 degrees Celsius for 2 minutes. Vescadine, an iodophore disinfectant diluted at 1 to 230, or combined beta-propiolactone. Lastly, UV radiation. The UV rays reach to the genetic material and induces dimerization of nucleic acids to prevent virus from replicating.